In this program, we look into the meanders of memory, the sources of laughter and tears, and the mysterious realm of aptitudes. Neuropsychology is the integrated study of neurology, science of the brain, and psychology, science of behavior. Neuropsychology attempts to solve a baffling puzzle. How can an organ the size of a grapefruit manage to engender emotions, thoughts, and memory? Learning to play a musical instrument. Remembering one's first trip. Retaining a telephone number by heart. These are all tasks that depend on memory. In recent years, scientists have begun to unravel the processes involved in memory functions. They have discovered that there are several types of memory, and that each one involves a different part of the brain. For instance, retaining a telephone number we've just heard is a task related to short-term memory. Short-term memory has been localized in the parietal lobe of the cortex, a part of the brain surface characterized by deep folds. It allows us to assimilate information for a brief moment, roughly 1 minute. We often use short-term memory, for example, to retain new information useful in carrying out a task. Normally, this information is quickly forgotten. However, if frequently reused, it may be transferred to our long-term memory for more permanent storage. Long-term memory allows us to preserve information for years, even our whole lives. There are 3 types of long-term memory. First of all, events in our personal lives are retained by episodic memory. Each event stored in this memory is usually associated with a specific date and place. Recent research has shown that the hippocampus, located beneath the cortex, and the thalamus, buried in the center of the brain, play an important role in episodic memory. It appears that at birth, these structures have not yet reached full maturity. This would account for the vagueness of our childhood memories. When we learn to play a musical instrument or to drive a car, we depend on procedural memory. This memory enables us to acquire various skills, either through practice or repetition. Once we have mastered these skills, they become almost automatic. Procedural memory involves part of the frontal cortex, as well as a structure nestled under the cortex, the gray matter nuclei. The third form of long-term memory encompasses what we know of the world and our general knowledge. It is where we store rules of grammar, laws of arithmetic, or words. The operations of this type of memory are carried out in various regions of the cerebral cortex. Whenever information is imprinted on any one of the 3 forms of long-term memory, it engraves a mark which, in a sense, constitutes a memory. That mark corresponds to a modification of the nerve cells. Let's take the example of an actor preparing for a role. First, he acquaints himself with the various aspects of his performance. His brain creates internal representations of the scenes. Each of these representations monopolizes the activity of a specific network of nerve cells. Indeed, the brain is made up of billions of nerve cells called neurons. These neurons communicate with each other by releasing chemical messages at the sites where they connect. As our actor continues to rehearse, these points of contact between the various neurons involved consolidate. This enhances the transmission of information between the neurons. The strengthening of these connections leads to the actors committing to memory the monologue and gestures he will be performing during the show. But how do the nerve cells manage to retain information? To find out, researchers studied the basic mechanisms of memory in a small marine snail, Euphia. This animal is used in studies because its simple nervous system consists of relatively few giant-sized neurons. The researchers found that Euphia can learn to ignore a stimulus when it is repeated several times. Normally, it will withdraw its gill as soon as a stimulus is applied to another part of its body, such as the mantle shelf. But when the stimulus is applied repeatedly, its withdrawal reflex gradually diminishes. The researchers noted that following this conditioning, the connections between the neurons underwent structural changes. They observed, notably, a decrease in the number of synaptic knobs contained in the neurons' message-bearing chemicals. Communication between the neurons is thus modified. These findings in Euphia show that in long-term memory processes, the structure of nerve cells change. According to researchers, it is highly probable a similar phenomenon occurs in humans. For instance, when the actor learns his text, he repeats it several times, enabling the neurons to modify their connections permanently. This would account for the fact that the older a memory is, the less likely it is to be forgotten. Indeed, the more often we recollect a memory, the more fixed it becomes in our long-term memory. Our nerve cells therefore bear the indelible marks of our childhood memories. It was recently discovered that emotions could be explained by the electrical and chemical activity of certain areas in the brain. Neurotransmitters, the messengers of the nerve cells, seem to play an important role in this biology of emotions. They would be responsible for spurring laughter, sadness, and fear. Any situation will give rise to emotions that are expressed throughout our bodies. Before they become manifest outwardly, emotions begin in the brain. They are, however, often brought on by external stimulation. For instance, when people watch a clown performing, various regions of their cerebral cortex receive information. This information triggers bursts of nerve impulses in the cortex. These impulses are relayed to the limbic system, a collection of structures in the brain involved in emotional behavior. They induce an affective sensation of joy and pleasure. If the emotion is sufficiently intense, the limbic system will pass the information on to a neighboring structure, the hypothalamus. It is the hypothalamus that expresses the emotion through the body. In our example, it will induce laughter. The physical expression of laughter sets in motion numerous muscles throughout the body. Just in the face, no fewer than a dozen muscles are involved, not even counting the neck muscles. In addition, messages reach the muscles of the rib cage and the diaphragm. The contraction of these muscles induces the spasmodic breathing that characterizes laughter. When they contract, the muscles expel air out of the respiratory tract at a speed of about 100 kilometers an hour. The pain caused by their contracting will make the person hold his sides. In most cases, the stomach muscles are also involved. When the stomach muscles contract, the pain can be so acute, the person will double over in laughter. These spasmodic expirations lead to a scarcity of oxygen in the muscles. The heartbeat then quickens to make up for it. If the person can't stop laughing, his leg muscles will give out and he may collapse. Later, as the laughter gradually subsides, the person is left with a wonderful feeling of relaxation. This feeling of well-being is attributed to the abundance of certain neurotransmitters, the nerve cells' message-bearing chemicals released in the brain during laughter. The coming back to normal of the heartbeat and muscles also contribute to this overall feeling of serenity. Conversely, a tragic event of painful memory can bring on a feeling of sadness. This emotion also develops in the limbic system. However, the frontal lobes of the cerebral cortex can retard the triggering of the limbic system and therefore the expression of sadness. This area of the cortex, located in front of the brain, plays a key role in the voluntary control of the expressions of feelings, whether sad or happy. The frontal cortex allows a grieving person to contain their sorrow. However, if the pain is too acute, the limbic system will notify the hypothalamus, which then commands the triggering of various outward signs. For instance, the muscles of the pharynx contract, causing the characteristic tightening of the throat. The lacrimal glands are stimulated and produce tears. In addition, breathing becomes halting, and the facial muscles contract, communicating an expression of despondency and despair. Together, these outward signs are a cry for help, and they do draw the attention and caring of the people who witness them. Important outward signs also occur when we are in danger. Accompanying the feeling of panic is an accelerated heartbeat. The heart pumps more oxygen and glucose to the muscles. Blood circulation temporarily curtails its irrigation of the digestive system to provide greater nourishment to the muscles. Breathing becomes shorter and faster, making for greater oxygenation of the blood. The face tenses in fright. The muscles tense up as well, in preparation for the fight or flight response. Feeling sad, frightened, or anxious is not harmful in itself. Rather, it is a normal reaction to stress. The loss of a loved one causes us to withdraw from the world for the time we need to mourn. Actually, moderate anxiety will often give us the determination to overcome illness, job loss, or marital difficulties. The intensity of emotional reactions varies considerably from one person to the next. However, although sometimes the body language is very different from one culture to another, the outward signs associated with the various basic emotions, such as gesturing, voice inflections, facial expressions, are universal. This only confirms that feelings are fixed in our genes and governed biologically. Intelligence is another infinitely complex ability. In this area, research is only in its infancy. The neuropsychologists are still developing instruments to define and measure it. The various approaches work well, but no one really knows what it is they're measuring. Humans possess amazing abilities. The number of skills and complex tasks that we manage to learn thanks to our intelligence is simply astounding. These abilities are ascribed to the refinement of the brain's structure and organization. To understand them better, neuropsychologists have developed various measuring devices. For instance, Faces are reconstructed according to a very complex multidimensional plan. Their appearance changes according to emotional stances and expressions, and they differ from each other only very subtly. However, our brain allows us to easily recognize someone we have met before. In order to better understand this ability and to define the role of the two hemispheres of the brain, researchers use a tachydoscope, a variable speed shutter device. This device allows the operator to present visual information to a single hemisphere. An image flashed on the far right-hand side of the visual field will be relayed to the left hemisphere of the brain, and vice versa. This is because the optic nerves leading from the eyes to the brain are crossed. During this experiment, the subject is asked to fix his gaze on a central spot, while images flash on one or the other side of the screen. If the image flashes in the most lateral part of the subject's visual field, it will be relayed only to the hemisphere on the opposite side. The device is also equipped with a camera that detects eye movements and allows the tester to make sure the subject's eyes are fixed on the central spot of the screen. It is important his eyes do not move, for this would allow the visual information to reach both hemispheres instead of just one. This method has enabled researchers to show that the left hemisphere detects details, such as a beauty spot, better than the right. Our left hemisphere therefore helps us to distinguish between two very similar faces, like those of twins. Conversely, the right hemisphere deciphers the overall configuration of a face, how the main features are organized, for instance, the distance between the eyes, or between the nose and the mouth. Most of the time, it is the right hemisphere that allows us to recognize a face, especially when it is far away. Recognizing faces is a complex skill, but intelligence is even more so. Innumerable tests have been developed to measure this set of faculties. One is the intelligence quotient test. The IQ test attempts to evaluate the variability of certain intellectual performances within a given group. It consists of two series of tests. The first consists of oral tests. The examiner checks the subject's general knowledge, practical judgment, and short-term memory by making him repeat a series of numbers backwards, for instance. He also measures his ability to reason logically by asking him to identify the similarities between different objects. This series of tests also assesses the subject's ability to solve mathematical problems and to define words. The series consists of non-verbal tests, which are timed. They evaluate the subject's motor skills and patterning of space and time. They involve situations, for instance, in which the subject has to put a series of images in the proper order to recreate the chronology of a story. In another test, the subject is asked to identify the details missing from a picture for it to be coherent. The final tests consist of putting a jigsaw puzzle together and reproducing a geometric shape with blocks. The grading is done by awarding 1 or 2 points for every test completed successfully. The intelligence quotient is calculated by comparing the subject's score against the one set by an average of persons of the same age. The median IQ is arbitrarily set at 100, and the majority of people achieve scores varying between 85 and 115. A small minority of 2% achieve an IQ of 130 to 140. Another group scores between 30 and 85. But curiously, in the past 30 years, the results of IQ tests have shown improvement in the industrialized nations. In one generation, the average IQ has jumped 15 points, While intelligence is the combined product of heredity and environment, this increase is believed to be due to environmental factors. The IQ test was originally designed to predict a child's ability to succeed in school. However, it has been used abusively, for example, as a way to compare races. Yet it is generally agreed that the IQ test only assesses some aspects of the intellectual process. It chiefly analyzes abstract intelligence, that is theoretical, logical, and formal intelligence. The IQ test still has its uses, but according to researchers, it is not sufficient to measure intelligence as a whole, and much less to define it. Intelligence derives from the effect of our environment and our activities, together with that of our genes. As Thomas Edison once said, genius is 1% inspiration and 99% perspiration.